Hello dear students, how are you all? This is Praful and I welcome you all to my YouTube channel, that is One Page Biology. Dear students, in today's video we are going to study another very important topic of biology, which is nothing but structure of DNA. Dear students, this particular topic is part of 12th standard NCERT biology chapter Molecular Basis of Inheritance, and this chapter is from unit number 7, which is nothing but genetics and evolution. So are you all ready for this particular topic, dear students? So let's begin with the video. So dear students, basically where exactly is DNA located? First of all, DNA is basically nothing but a nucleic acid. The full form of DNA is deoxyribonucleic acid. And we all know that DNA is located inside the nucleus of the cell. Yes, dear students, nucleus, which is nothing but the brain of the cell, which controls all the different functions of the cell. So who exactly discovered this particular nucleus? Dear students, Friedrich Mischer had discovered a particular substance which was found in the nucleus of the WBCs of pus cells. Now Friedrich Mischer had named such a substance as nuclein, and later on it was found that this particular substance called as nuclein had acidic properties, and that's why this particular substance called as nuclein was given the name nucleic acids. So today, dear students, we all know that there are two types of nucleic acids. One is DNA and the other is RNA. DNA stands for deoxyribonucleic acid, whereas RNA stands for ribonucleic acid. So dear students, DNA is present in the form of chromatin threads which are present inside the nucleus. These chromatin threads are in highly condensed form, which are known as chromosomes. So these chromosomes contain the DNA along with some very important proteins which are known as the histone proteins. Dear students, DNA is extremely important because DNA controls all the characteristics of the particular organism, and that's why DNA is termed as the genetic material of the cell. Now dear students, back in the year 1953, the very famous scientists, that is James Watson, Francis Crick along with Rosalind Franklin, had discovered the double helical structure of the DNA. Yes dear students, according to this particular model, DNA is nothing but a double helical structure. That means DNA consists of two particular strands which twist around themselves. And for this, dear students, James Watson, Francis Crick and Rosalind Franklin received a Nobel Prize for the same. Now let us understand the double helical structure of DNA, which was discovered by James Watson, Francis Crick and Rosalind Franklin. Now according to them, DNA is basically a double helical structure. Double helical means it consists of two strands which basically twists around themselves. At the same time, dear students, DNA, which stands for deoxyribonucleic acid, is made up of many nucleotides. That's why, dear students, it is referred as a polynucleotide chain. So dear students, as we said that DNA consists of two strands which are coiling around each other. Now these strands are made up of nucleotides. And what exactly is a nucleotide? Dear students, nucleotide is basically a type of biomolecule which consists of three main components. These three components are nothing but a 5-carbon sugar, which is basically either a ribose sugar or a deoxyribose sugar. So dear students, in case of DNA, it is always a deoxyribose sugar which is part of the nucleotide. Apart from the 5-carbon deoxyribose sugar of a nucleotide, the nucleotide consists of other two very important components. One is nothing but the nitrogen base, which are of basically two types, purines and pyrimidines, and the third main component is nothing but a phosphate group, which is also attached to the sugar. Now dear students, as we just said that a nucleotide consists of three main components, which are nothing but a 5-carbon sugar, a nitrogenous base as well as phosphate group. Now dear students, remember that the nitrogenous base is always attached at the first carbon of the sugar, whereas the phosphate group is attached at the fifth carbon of the sugar. Now dear students, this sugar, which is primarily of two types, one is ribose sugar, the other is deoxyribose sugar. So what is the difference between ribose sugar and deoxyribose sugar? The difference between ribose sugar and deoxyribose sugar is basically the deoxyribose sugar does not contain an oxygen atom which is at the second carbon, whereas the ribose contains an extra oxygen which is present at the second carbon. Now dear students, we should know that the nitrogen base which is connected to the sugar of a nucleotide is basically attached by a bond which is called as glycosidic bond. Now, as far as nitrogenous bases are concerned, dear students, there are two basic types of nitrogenous bases. One are nothing but purines and the second are pyrimidines. What is the difference between purines and pyrimidines? Remember, dear students, purines are basically double ring compounds, whereas pyrimidines are always single ring compounds. Purines are basically of two types, which are nothing but adenine and guanine, whereas pyrimidines are of two types, which are basically cytosine and thymine. Dear students, in case of RNA, thymine is replaced by another type of pyrimidine which is basically called as uracil. Now dear students, as you can see that according to the Watson and Crick model, the two strands which are coiling around each other are basically connected to each other. These two strands are basically connected by the nitrogen bases on the opposite strands, and the nitrogenous bases are connected to each other via the bonds which are nothing but the hydrogen bonds. Now in this particular diagram, as you all can see, that purines are getting connected to the pyrimidines. That is, always adenine is pairing with the thymine, whereas guanine is pairing with the cytosine. At the same time, dear students, between the adenine and the thymine there are always two hydrogen bonds, whereas between guanine and cytosine there are three hydrogen bonds. Now dear students, as per the Watson and Crick model, one of the most important features of the DNA double helical structure is basically both the strands are complementary in nature. That means, dear students, always purines are paired with the pyrimidines. That means always, dear students, adenine pairs with thymine, at the same time guanine pairs with cytosine, or vice versa. There was one very important scientist whose name was Erwin Chargaff, who came out with a very important rule. This rule was called as Chargaff's rule, which states that always in the DNA the total number of purines are equal to the total number of pyrimidines. In other words, dear students, the total number of adenines will be equal to the total number of thymines. At the same time, the total number of guanines will be equal to the total number of cytosines. Or else we can also say that A plus T upon G plus C will be equal to 1. So dear students, this is very famously called as the Chargaff's rule. Now dear students, another very important feature of DNA double helical structure is both the strands are antiparallel in nature. That means both the strands are opposite in direction. So that means, dear students, if one strand has a direction from 5 prime to 3 prime, the other strand will have the direction from 3 prime to 5 prime. Dear students, remember that the nucleotides are connected to each other by very important bonds which are known as phosphodiester bonds. This is a kind of an ester linkage. That's why, dear students, the phosphoester linkages are also considered as the backbones of the DNA. It is because of these phosphoester linkages, dear students, the structure of DNA remains stable. Now let us understand certain dimensions of the DNA. Now dear students, first of all, the diameter of the DNA is around 20 Armstrongs. Remember, 1 Armstrong is equal to 10 raised to minus 10 meters. So in other words, the diameter of DNA, which is 20 Armstrong, can also be denoted as 2 nanometers. Remember that because of the twisting of the two strands around each other, there are two major gaps which are created. One is a smaller gap and the other is a larger gap. The smaller gap is called as a minor groove, whereas a larger gap is called as a major groove. The minor grooves and the major grooves are present one after the other. Now dear students, the length of a single turn is around 36 Armstrongs. At the same time, dear students, the distance between the two consecutive base pairs is basically 3.4 Armstrong. Now what do we mean by a base pair? A base pair is nothing but the two nitrogen bases which are connected to each other. Likewise, the distance between the two consecutive base pairs is 3.4 Armstrongs. This is known as the pitch of the DNA. Now dear students, there are 10 base pairs in a single turn. So dear students, that was about the structure of DNA. If you have understood the concept of DNA, please like the video. Also do share with your friends, dear students. If you have any doubts or suggestions, please mention them in the comment section. So see you all in the next video with some another biology related topic, dear students. Take care of yourself. This is Praful signing off. Thank you so much. Bye bye.